Hey guys, how's it going? This is FixerMed. Welcome back to my high yield genetics review series for the USMLE Step 1, NBME CBSE, and NBME CAS examinations. This will be part 5 of my multi-part video series covering the entire discipline of genetics in a high yield but comprehensive manner. So without further ado, let's go ahead and get started on today's content. Today is going to be a relatively dense lecture because it is molecular genetics and a lot of this stuff is super high yield and builds up in the biochemistry discipline as well. So let's go ahead and get started by talking about the DNA structure and replication. So DNA structure, mainly made up of a double helix structure. DNA consists of two polynucleotide strands bound around each other in a helical shape, forming a double helix. The nucleotide components, each nucleotide includes a phosphate group, a sugar molecule, also known as deoxyribose, and one of 4 nitrogenous bases, adenine, thymine, cytosine, and or guanine. Complementary base pairing, adenine pairs with thymine via 2 hydrogen bonds, and cytosine pairs with guanine via 3 hydrogen bonds. This complementary base pairing ensures stability in the DNA double helix. DNA structure has an anti-parallel orientation. The 2 strands of the DNA molecule run in opposite directions, with one strand oriented in the 5 to 3 direction and the other in the 3' to 5' direction. Now let's go ahead and start talking about the DNA replication process. DNA replication is a semi-conservative process where the parental DNA molecule unwinds and separates into 2 strands. Each strand serves as a template for the synthesis of a new complementary strand. Unwinding, the DNA double helix is unwound by helicase enzymes, which break the hydrogen bonds between the base pairs, creating a replication fork. Template strand, each separated DNA strand serves as a template for the synthesis of a new complementary strand. DNA polymerase reads the template strand in the 3' to 5' direction and synthesizes a new DNA strand in the 5' to 3' direction. Leading and lagging strands, DNA replication occurs bidirectionally at the replication fork. The leading strand is synthesized continuously in the 5' to 3' direction, while the lagging strand is synthesized discontinuously in short fragments called Okazaki fragments. Role of DNA polymerase. DNA polymerase catalyzes the addition of nucleotides to the growing DNA strand using the parental DNA strand as a template. It also possesses proofreading activity to ensure the accuracy of DNA replication. Fidelity of DNA replication. DNA replication is highly accurate due to the fidelity of DNA polymerase and the proofreading mechanisms. DNA polymerase has an intrinsic 3' to 5' exonuclease activity, allowing it to remove mismatched nucleotides and replace them with correct ones, thus minimizing errors in DNA replication. Moving on. Next, we're going to be talking about RNA transcription and translation. RNA transcription, to give a brief overview of the process, we have 3 steps: initiation, elongation, and termination. Transcription begins with initiation, where RNA polymerase binds to the promoter region of DNA. During elongation, RNA polymerase reads the DNA template and synthesizes RNA in the 5' to 3' direction. Termination occurs when RNA polymerase reaches a terminator sequence, leading to the release of the newly synthesized RNA molecule. Different types of RNA include mRNA, tRNA, rRNA. mRNA's main role is to carry genetic information from DNA to the ribosome for protein synthesis. tRNA, known as transfer RNA, brings amino acids to the ribosome during translation, matching them with the codons on mRNA. rRNA, ribosomal RNA, is a component of ribosomes where protein synthesis occurs, providing structural support and catalyzing peptide bond formation. Then we have RNA translation. Similar steps: initiation, elongation, termination. Translation begins with initiation, where the small ribosomal subunit binds to the mRNA molecule. During elongation, amino acids are added to the growing polypeptide chain according to the codons on the mRNA, facilitated by tRNA molecules in the ribosome. Termination occurs when a stop codon is reached, leading to the release of the completed polypeptide chain. The role of ribosomes, tRNA, and amino acids in protein synthesis. Ribosomes are the cellular machinery where protein synthesis occurs, providing the site for mRNA and tRNA interaction. tRNA molecules carry specific amino acids to the ribosome, where they are added to the growing polypeptide chain based on codon-anticodon recognition. Amino acids serve as the building blocks for proteins, forming peptide bonds to create the primary structure of the protein. Central dogma: highlight the flow of genetic information from DNA to RNA to protein. The central dogma of molecular biology describes the sequential flow of genetic information. DNA is transcribed into RNA through the process of transcription, and RNA is translated into protein through the process of translation. The unidirectional flow of information ensures the transfer of genetic instructions from DNA to functional proteins, essential for cellular processes and organismal development. A lot to take in there, but moving on. Genetic code, codons and anticodons. The genetic code is defined by codons, sequences of 3 nucleotides on mRNA, each coding for a specific amino acid during protein synthesis. The relationship between mRNA codons and tRNA codons is complementary, ensuring the accurate alignment of amino acids during translation. The start and stop codons. The start codon is known as AUG, which initiates protein synthesis, marking the beginning of translation. The stop codons include UAA, UAG, and UGA, signal the termination of translation, ensuring the proper completion of the polypeptide chain. And I don't know if you guys recall this, but in the biochemistry review series, I gave a good way to remember the stop codon. It's kind of ridiculous, but I'll go ahead and do it again. It's UGA, UAA, and UAGA. For some reason, those sounds stick to me, and every time I hear stop codon, I just hear UGA, UAA, and UAGA. So I hope that helps you guys out too. And start codon, I've just memorized AUG as methionine at this point because I've seen it so much. So I hope you guys are able to recall this as easily as well. Moving on. Mutations, types and consequences. There are multiple types of mutations. There are point mutations, which include substitution, insertion, and deletion, which involve a change in single nucleotides or small segments of DNA, while frame shift mutations alter the reading frame of the genetic code, potentially leading to significant downstream changes. Silent mutations have no observable effect on the resulting protein. Missense mutations result in the substitution of one amino acid for another, and nonsense mutations introduce a premature stop codon, which shortens the protein. Truncating just means shortens. Moving on, we have the consequences of mutations. Mutations can disrupt the structure and function of proteins, which lead to altered cellular processes or malfunctioning biological pathways, and they serve as the underlying causes of various genetic diseases and contribute to evolutionary changes in populations over time. Repair mechanisms, DNA repair mechanisms, including base excision repair, nucleotide excision repair, and mismatch repair, serve to identify and correct errors in DNA sequences, which play a crucial role in maintaining genomic integrity and preventing the accumulation of mutations that could otherwise lead to harmful consequences. Moving on. Next, we have questions pertaining to the material that we discussed. A lot of material, so if you guys need a quick break, just go ahead and pause the video. I'm just going to go ahead and start the questions, though, for anyone that just wants to go right into it. So we have a patient that presents with a genetic disorder characterized by an autosomal recessive inheritance pattern. Pedigree analysis reveals that both parents are carriers of the mutated allele but are phenotypically normal. Which of the following is the most likely genotype of the affected individual? Pretty first order question, review question from the first genetics review presentation. So if you need more time, please feel free to pause the video. Otherwise, moving on now. In an autosomal recessive inheritance pattern, affected individuals are homozygous for the mutated allele. Little a, both parents of affected individuals are typically heterozygous carriers, big A, little a, and are phenotypically normal. When two carriers, big A, little a, have children, there is a 25% chance with each pregnancy of having an affected child. little a, a 50% chance of having a carrier, big A, little a, and a 25% chance of having an unaffected child, big A, big A. Therefore, the most likely genotype of the affected individual is homozygous for the mutated allele, little a, little a. Therefore, the correct answer is C, little a, little a. Moving on to see why the other answer choices are incorrect. Big A, homozygous dominance would result in a phenotypically normal individual. This genotype is not consistent with an autosomal recessive inheritance pattern. Big A, heterozygous genotype represents carrier status. While carriers may pass on the mutated allele to their offspring, they do not exhibit the phenotype of the genetic disorder. Big A, big B, little b. This genotype is not relevant to autosomal recessive inheritance. It represents a hypothetical dihybrid cross with incomplete dominance, which does not apply to the scenario described. Big A, big B, homozygous dominant genotype, big A, big B would result in a phenotypically normal individual. This genotype is not consistent with an autosomal recessive inheritance pattern. Moving on to the next question. A researcher is studying a genetic disorder caused by a mutation in a gene located on the X chromosome. This disorder affects both males and females, but males typically exhibit more severe symptoms. Which of the following inheritance patterns is most likely associated with this disorder? Pretty first order review question, so if you need more time, pause the video. If not, moving on now. The inheritance pattern described where males are more severely affected and both males and females can be affected suggests X-linked recessive inheritance. In X-linked recessive disorders, affected males inherit the mutated allele from their carrier mothers. As males only have one X chromosome, females typically need to inherit 2 copies of the mutated allele, one from each parent, to exhibit the phenotype, while males need only 1 copy. Therefore, males are more likely to be affected and exhibit more severe symptoms. Therefore, the correct answer is D, X-linked recessive. See why the other answer choices are incorrect. Autosomal dominant disorders typically affect males and females equally, and affected individuals typically have an affected parent. The inheritance pattern described does not fit the characteristics of autosomal dominant inheritance. Autosomal recessive disorders typically affect males and females equally, and affected individuals often have unaffected carrier parents. The inheritance pattern described does not fit the characteristics of autosomal recessive inheritance. X-linked dominant disorders typically affect both males and females, but affected males do not usually exhibit more severe symptoms than affected females. Additionally, affected males must pass the mutated allele to all of their daughters, as all daughters inherit their X chromosome from their father. The inheritance pattern described does not fit the characteristics of X-linked dominant inheritance. Y-linked disorders are passed only from father to son and typically affect males exclusively. The inheritance pattern described involves both males and females being affected, so Y-linked inheritance is not applicable. Guys, that will do it all for today's content. And if you guys are wondering why the questions seem to be pretty basic, the main bulk of this content will actually be more pertinent in the biochemistry, molecular, and cell biology section. I think it's part 1 of that video series, so if you guys want a bigger rundown of the details of mutations, genetic code, RNA transcription, and translation, you'd be better served finding that in the biochemistry review series for practice problems purposes. This is more so a broad overview of everything you need to succeed on those type of questions. Just basically went into the weeds a little bit more, so I hope this helped you guys out. And if you guys have any questions, comments, or concerns, please feel free to leave them in the comment section below. Be sure to leave a like and subscribe for more content. Anyways, this is FixerMed signing off. Good luck studying and goodbye, everyone.